Welcome to the Syneos Health Podcast, Early Signals, where we explore the ideas, innovations, and emerging trends shaping the future of oncology. I'm Dr. Wael Harb, Head of R&D and Scientific Strategy in Oncology at Syneos Health. Recorded live from ASCO 2026, these conversations bring together perspective from leaders across biopharma, academia, and technology on the signals they're watching most closely, from AI-enabled drug development and real-world evidence to precision medicine. These conversations offer a snapshot of where oncology innovation is headed next. Let's tune into one of our conversations now. For decades, MYC has been called oncology's greatest white whale, a transcription factor implicated in the majority of human cancers, a driver across nearly every hallmark of cancer, and yet stubbornly resistant to direct small molecule targeting because of its disordered structure and lack of an obvious binding pocket. Is a target everyone agrees matters and almost no one has been able to drug. My guest today is taking that challenge head-on. Nick Glover is CEO of MycRx, a company developing oral small molecules designed to directly engage MYC on DNA and shut down its oncogenic transcriptional program. Nick brings a remarkable operator's track record to this. He led Sierra Oncology and YM Biosciences, the latter through its acquisition by Gilead, so he knows what it takes to move a hard idea from discovery into the clinic. Nick, welcome to Early Signals.
Thanks, Wael. It's a pleasure.
MYC has been described as undruggable for decades. For listeners who don't live in this biology day to day, why has it been so hard, and what made you believe it was finally targetable?
Now, that's a great question. It's the key question with MYC. The analogy that makes most sense is RAS. For a long time, we thought that RAS was undruggable. Clearly, we walk around ASCO today, we look at the amazing success of the G12Cs and now the pan-RAS inhibitors, RevMed's amazing data in pancreatic. It's clear that RAS is a druggable target. What opened up the druggability was structure-function relationships, and understanding how to design small molecules against the target. The generally held wisdom that MYC is undruggable because it lacks structure is clearly not correct. MYC has to engage on DNA in order to do what it does biologically, which is to drive transcription. Clearly, it has to engage with DNA in a structured manner. It really is a case of understanding when and how MYC becomes structured, to further deconvolute what that means in terms of identifying a druggable site, and then having the ability to design clever small molecules that can take advantage of that druggability. So our breakthrough really starts with structure. Being able to understand how and where MYC adopts a three-dimensional structure that is amenable to small molecule design, leveraging and exploiting that design in a concerted chemistry program that's oriented towards leveraging that structural understanding, and then obviously furthering that with a series of mechanistic biology and SAR-driven assays that lead you towards the delineation of a small molecule that has the properties that are capable of being a drug. And that's something that we've taken a long time to figure out. There were original discoveries at the company prior to my involvement that led us to believe truly that the chemical matter that the company had was engaging MYC. And using that chemical matter essentially as the bait, we went fishing for what that structure looked like. We were able to determine exactly where our compounds were binding to MYC. We were able to use a suite of structural biologic assays to further delineate with great specificity the particular amino acid residues on MYC that our compounds were engaging with. And then the rest is obviously a case of systematically building chemical matter that's potent, is selective for MYC, and obviously has the requisite pharmacologic properties required to become a drug. And that wasn't a trivial exercise in and of itself. It took several years, even following our, I think, seminal breakthrough discoveries of the structural information required to enable small molecules, to actually be at this point today, which I'm actually delighted to announce, that we do have a drug candidate. We've just nominated our drug candidate. We're ready to move into IND-enabling studies. And we expect to be starting clinical studies essentially a year from now, around mid-2027, we'll be in the clinic with what we believe is the first true direct small molecule inhibitor that directly binds to MYC and directly perturbs MYC's oncogenic transcriptional biology. So we're very excited about that prospect and look forward to further talking about it this morning.
That's a great analogy. RAS has been considered undruggable for decades, and now, especially at this conference, we have a great release on great advances against RAS in very difficult to treat cancer, pancreatic cancer. With MYC, for our audience who have not heard of MYC, can you tell us why MYC is important for the biology and how many patients that tumors are affected by MYC?
That's a fantastic question. And maybe I'll just take a step back to talk a little bit about myself, if I can. Like a great many people here at ASCO, cancer isn't just a job. It just isn't something that we've focused our careers on. For many of us, and for me particularly, cancer's personal. Cancer is something that hits under your roof, it hits within your families, it hits within your friends, and it affects each and every one of us. And I feel very passionately about trying to help as many patients as possible who have cancer, and I have done throughout my career. It's always been my North Star to think about how can the drugs I'm involved in developing really make a difference, and to focus on things that were differentiated, things that offered a chance to achieve a benefit or a safety outcome that other agents hadn't achieved. Obviously, that led me to MYC. Now, MYC is a target that's critically important, and I think the truism is MYC is generally important for every cancer. Now, that seems like a very bold claim, but let me reinforce why I think we can stand behind that. We've looked in real-world data, so actual patient data, and we've looked to understand across every cancer, whether that's a solid tumor, whether that's a hematologic malignancy, whether MYC biology was relevant to that background. And we've worked with one of the key vendors in the real-world data space to analyze about 125,000 patients to understand MYC biology. And it's given us a very clear roadmap. But the take-home message is there are MYC-sensitive populations in every cancer, every solid tumor, every hematologic malignancy. So MYC is generally important to every cancer, but in some cancers, it's really, really important. And it's a case of understanding the differences between those backgrounds in which MYC is part of the engine room, and in those backgrounds in which MYC is the principal or predominant driver of the oncogenic process. So what does MYC do, and why is MYC important in cancer? Well, MYC is essential. MYC is an essential gene. MYC is an essential protein driving transcription in normal cells. It has key functional roles in driving things like hematopoiesis. It is involved in the ordinary process of cellular proliferation. It's an essential element in normal cells. The challenge with MYC in oncology is that MYC is frequently found to be overexpressed. So you have an abundance of MYC. In many cases, you have a significant excess of MYC. When there's a lot of MYC in cancer cells, it essentially acts as a bully. What it does is it crowds out on DNA different promoters, and it sits on those promoters, and it drives the transcription of genes that ordinarily it wouldn't be transcribing. And so essentially it co-opts the transcriptional machinery in cancer, and it drives the output that in turn drives the biology that is essentially, as you already remarked, that is the hallmark of what makes cancer cancer. So MYC, by sitting on transcriptional start sites that it typically shouldn't be sitting on, drives a whole series of biology that are involved in processes like proliferation, cell cycle, the management of the immune response to cancer, how cancer deals with genomic instability and mutation, how it deals with angiogenesis. All of those biologic processes that have been codified in what we describe as the hallmarks of cancer, essentially a rubric for understanding the various classes of biology that have become associated with cancer. MYC is at the center of all of that biology. So not surprisingly, MYC is generally important. But in some cancers, MYC is really the predominant driver of all of that biology. In other cancers, there are other oncogenes, there are other mutations that also contribute But in many, many cancers, MYC is the most important thing, or one of the most important things that's happening to drive that oncogenic process. So not surprisingly, the opportunity therapeutically for MYC inhibition is enormous. Not just as a monotherapy. Clearly, if you can identify patients whose disease is strongly, strongly driven by MYC, then that's a great place to go as a monotherapy. But because MYC is so important generally, the prospects for combination therapy are incredibly broad. Again, because of the underlying biology that MYC is driving. I'll give you a couple of examples. So let's take non-small cell lung cancer, sort of a timely topic today. We have data coming out in the field here at ASCO around a new class of drug in non-small cell lung cancer, PD-1 VEGF bispecifics. It's been termed immunotherapy 2.0. It's a next generation of PD-1, PD-L1 inhibition combined with activation of angiogenesis. And those two things in combination, these bispecifics that many companies have, the data today was from Akeso. It's an asset that's been developed in North America by Summit. Shows an impressive response in first-line squamous cell carcinoma in non-small cell lung cancer. Potential to redefine a whole class of biology. So in that case, you've got two different therapeutic areas coming together, two classes of biology coming together in a therapeutic. One is obviously PD-1, PD-L1, the immune response, and looking to activate the immune response in cancer. And the other is angiogenesis, the VEGF component. Well, it turns out that MYC sits at the middle of both of those classes of biology. And so the opportunity for MYC to be a synergistic complement to both PD-1, PD-L1 therapy and VEGF therapy, or agents that combine both, is an obvious place to go as you think about how MYC could be combined. Similarly in RAS that we've already mentioned. The RAS pathway is a pathway that's strongly associated in the literature with MYC upregulation. And so the prospect for MYC to be exploited both as a way to deal with the inevitable resistance that you see to KRAS-driven therapy, or potentially in combination to deepen responses and potentially to obviate the resistance mechanisms from occurring in the first place, given that MYC is the pathway through which much of that resistance is actually affected. And so you've got an enormous breadth of opportunity. In fact, it's one of the challenges in our program is to triage through opportunity space and clinical space and think about the places where we would want to test a drug first in order to obtain evidence of activity, of safety, and to set up potential registration paths. And obviously, as we're working with a CRO like Syneos, is critically important that as we sort of think through those next steps in the program. But hopefully that answers your question as to the breadth and the potential opportunity for MYC.
Is the opportunity clearly very big, which raised the question how you decide which tumor to target to start with, and which one you think is going to be the most dependent on MYC?
The benefit that we have in modern oncology is the ability to synthesize information from a variety of new modalities, new tools and technologies that provide insights that we've never had previously. I'm a big advocate of real-world data. I'm a big advocate of multimodal data sets. I'm a big advocate of liquid biopsies and all of these enabling technologies that come to the fore. So as I mentioned, we've been working with one of the large companies involved in the real-world data space and looking to real-world data to identify those pockets of patients that are very, very strongly MYC-driven. And frankly, it's a roadmap. It enables you to delineate not just in a few patient samples, but quite literally in tens of thousands of patient samples, exactly where MYC biology is the principal driver or a key driver. So we can really segregate the oncology universe into places that are low MYC driven, that have moderate MYC drive, and those that are high MYC. So from a translational perspective, the obvious place to go initially is places that are high MYC. Some of those are high MYC, some of those are very high MYC. So I'll give you a few examples. I've already mentioned non-small cell lung cancer. In non-small cell lung cancer, the KRAS pathway, the RAS pathway, is strongly MYC-driven. That's a high MYC indication. The PD-1 pathway is a high MYC indication. In areas like CRC, colorectal cancer, and in triple-negative breast cancer. Triple-negative breast cancer, I think, is a fantastic example of how MYC biology evolves through the natural history of disease. And you can follow this, you can trace this evolution in the real-world data. So if you start with a patient who has hormone-responsive disease, a classic breast cancer patient. MYC is important in breast cancer, but the levels of MYC and the driving of MYC transcriptional biology in hormone-responsive breast cancer is on the lower side of the scale. As the patients progress, as they become hormone receptor not responsive, as they become either HER2 positive and then ultimately in that triple negative situation where they're no longer responsive to HER2 therapy, where they're not responsive to hormonal therapy, well, lo and behold, those patients are incredibly strongly driven by MYC. And it's really no surprise because MYC is the upregulated mechanism by which resistance to those biologic processes and the treatments that are affected against those biologic processes achieves resistance. You can find very readily in real-world data cohorts of patients and indeed entire sub-indications of patients that are MYC high. Those are the obvious places to go first. On the heme malignancy side, the prototypic MYC indication, the one that usually comes to mind when people think about MYC, is diffuse large B-cell lymphoma. These are tumors that have been long associated with upregulated MYC biology. These are diseases that, in general, have upregulated processes that impede apoptosis. Well, again, we see, from a biologic perspective, really quite remarkable effects on apoptotic biology in preclinical models that have been treated with our MYC inhibitor. In those kind of lymphoid backgrounds, apoptosis is essentially turned off. What our compounds do is immediately turn back on the genes that drive apoptosis. And you see it at the genetic level. One of the beauties of MYC biology is the ability, with an incredibly granular degree of resolution, to understand changes in transcriptional biology. That kind of fingerprinting is incredibly useful. And so we can see the classes of genes that are driving apoptosis, the ones that are preventing apoptosis from happening, the things that are impeding these ordinary course outcomes. We see that those genes are immediately switched off or switched on with MYC inhibition. So coming back to the hallmarks of cancer, we can follow each and every one of the sets of genes that drives the biology that underpins the hallmarks of cancer. And the genes that are upregulated in cancer get downregulated following MYC inhibition, and vice versa, those that are downregulated get upregulated on the other side. And so you can follow and trace how MYC biology could be relevant mechanistically, biologically, at the transcriptional level, and you can see it in the real-world data. So it does provide a very clear roadmap for the places to go, and it gives you a very clear roadmap from the perspective of understanding, from a pharmacodynamic perspective, from a PD perspective, the changes that are occurring as a consequence of treatment. So as we look to translate and to define, okay, we've treated this patient with our MYC inhibitor, do we see MYC engaged? Do we see the changes in MYC biology that you would predict to happen because of inhibition? And we absolutely will be able to follow that in the clinic, which is going to be very, very useful as we start to translate.
Fascinating roadmap, and clearly taking the scientific background and the biology into the clinic. My question you mentioned earlier regarding how MYC is really important for normal cell biology and for cell proliferation, for hematopoiesis. Would that create a problem with toxicity? How are you going to be able to find the right window for the drug?
Absolutely critically important. So look, the North Star in drugging MYC has to be chronic therapy. There has to be the aspiration for this new class of biology. I think MYC inhibition potentially represents an entirely new platform. It represents a new class of therapeutic agent. No one has had an agent that can directly control the transcription of the genes that drive cancer before. It potentially opens up a whole novel MOA. Fantastic, sounds fantastic. But of course, there's always a downside. The downside is always, what's the tolerability profile, what's the safety profile? Critically important, especially if you want to get MYC under control in cancer, but importantly, keep it under control. So the North Star there is not just about an acute period of treatment, because imagine MYC is the engine room in cancer. That engine is always running. So regardless of your ability to treat a defined tumor or a defined mass at any point in time, MYC as an engine driving cancer is still there in the background. So the goal is not just to deal with an initial response. The goal is to deal with that initial response, but to maintain that response, to keep MYC under control once you've got it under control. That sets a new bar in terms of the ask pharmacologically. You need to have an agent that's going to be well tolerated over an extended period of time, not just a short, sharp shock. It's not just a brief treatment window that aspirationally we need to achieve here. Now, MYC does have a normal role, as I said. I think that's very well established. So first and foremost, what does MYC do in normal cells and how does MYC act? Well, as we mentioned, it's involved in processes like hematopoiesis, it's involved in proliferative processes in general. So you would imagine that if you inhibit MYC too severely, you could end up with hematologic toxicity, with GI toxicity, the quintessential hallmarks of an anti-proliferative therapy. Of course, all those things are readily monitorable, but I think more importantly, it's about the differences between MYC in cancer and MYC in normal cells. MYC in normal has redundancy. There are a number of other transcription factors that can be involved in normal function and the normal regulation of those processes. But critically, there are significant differences between cancer and normal. Not only is stochastically there are massive excess of MYC in cancer versus normal cells, the actual transcriptional complex itself, the entity, that conglomeration of different entities that drives transcription, of which MYC is the anchor, is different between cancer and normal. Different components, different resonance times, different burst dynamics of transcription. All of those things can be teased out between cancer and normal. We liken it to a rheostat, like the volume dial on your stereo. In cancer, MYC is turned all the way up. That signal is blaring out. There's a significant noise, a cacophony of MYC. In normal cells, MYC is turned on and off very briefly. It's a short burst. It's a small window. It's a little bit of output, but then quickly turned off. So as you can imagine, you can exploit that difference, that ability to bring down the level of MYC in normal, to turn down the rheostat, to turn the volume dial down on MYC, down to normal levels. And you can do that by training your chemistry. And so we trained our chemistry with obviously an on-target structure-activity relationship for MYC in oncology, and we counter-train against MYC in normal. So MYC in normal peripheral blood, MYC in normal fibroblasts, MYC in its low, well-controlled, very short burst dynamic state, the normal MYC system. And what we've been able to do in technical terms is achieve very low, even sub-nanomolar potency in cancer, but retain high double-digit micromolar activity in normal. So the drug candidate that we've just nominated is incredibly potent in cancer, but is 20 micromolar in normal. It's a significant delta between the two. And what we've found in our preliminary animal modeling is that you can achieve a significant window between cancer and normal. If you can sustain a dosing, again in technical terms, that exceeds the therapeutic EC90, so the coverage of MYC at 90% levels, and sustain that around the clock, 24/7. But we can achieve that without seeing perturbation in the heme compartment, without seeing perturbation in the GR compartment. And I think it's one of the critically important things about drugging MYC, is to drug it directly. If you can drug MYC directly, and if you can affect its biology directly, then you get to calibrate and control exactly how hard you hit it. There's been a number of approaches tried over the years that have been indirectly trying to get at MYC. And what they've done essentially is shut MYC off completely. Well, of course, that's been incredibly toxic, because the normal function of MYC is turned off as well as the oncogenic function of MYC. And the collateral damage to that is enormous. And many of those things were not specific for MYC, of course, because they weren't directly binding MYC. And so they picked up collateral damage beyond MYC itself as well. And not surprisingly, those approaches, many of which have been trialed in the clinic, CDK9 inhibitors, bromodomain inhibitors, for example, different ways of getting at MYC through, say, for example, molecular glues against GSPT1, really not very well tolerated, and particularly not that active in terms of anti-cancer benefit. So we think by directly drugging MYC, by bringing down the levels of MYC, but not turning off MYC completely, you can sustain that attenuation of MYC's transcriptional output in cancer, but do it in a way that actually spares normal. And that's what we see in our preclinical data, and that's obviously something that we'll be testing as we go into the clinic. But that's the North Star here, to be able to have an anti-cancer benefit and sustain control over MYC without hitting the normal compartment.
That's fascinating. From a perspective of drug design and how your drug that you nominated and how it interacts with MYC, is that the way it interacts with MYC would have the differentiation with oncogenic MYC versus normal MYC? Or is it just the expression of MYC that really matters?
The reality is we directly hit MYC, but we hit MYC within the context of the complex that MYC exists in. Like all biology is complex in cancer, especially transcriptional biology is especially complex. And I say that both figuratively and literally. There is a complex of entities that arrange and array themselves around MYC as the central anchor. And you can exploit the fact that there are differences between that in cancer and normal in terms of the way that that modifies the binding site that we bind to. So yes, there's a stochastic difference, and that's essentially accountable for much of the difference between cancer and normal. But there are structural differences that are a consequence of the actual way that MYC is engaged on DNA in cancer and how it's engaged on DNA in normal that we also exploit. It really is taking advantage of directly drugging MYC in context of the way that MYC is found in cancer and being able to hardwire into the entire program, focus on that particular flavor of MYC versus the way that MYC exists in Renaissance.
Nick, you have vast experience in drug development and successfully taking drugs and companies to the next milestone and exit. As you are developing this novel MYC inhibitor, I noted that you already have exploring working with Australia. How do you think drug development in early phase oncology looking at from the outset as a global process?
I think first and foremost, every CEO of every company needs to recognize you're in the business of biotech. And the business of biotech is oriented towards, as we see here, we're looking out over the exhibit floor here at ASCO, looking at all the large pharma booths. Ultimately, drugs like this, especially a drug against MYC, needs to be in the hands of someone who has the capability, the breadth, the muscle to be able to take advantage of that both therapeutically and commercially. And so the end game in any biotech CEO's playbook is, how do I build a persuasive and compelling data set that enables me to catalyze a potential transaction with pharma, assuming the data all go your way? And that clearly is the end game for many, many companies. And certainly when you've got a franchise opportunity like this, clearly to capture the full opportunity, there's going to need to be a large entity involved at some point down the road. So as we think through the next steps, we've cracked discovery against MYC. Fantastic. I think it's an absolutely breakthrough achievement. But of course, you don't sit still. Now we need to think about, how are we going to develop this drug? Where are we going to develop this drug? And what are the key outputs that we need to achieve? And how are we going to achieve those things as quickly and efficiently as possible? So of course, job number one has to be, we need to understand a safe, well-tolerated dose as quickly as possible. And so our dose-finding work is key. And we need to establish that we do indeed engage MYC in the human system. Obviously, all of the work so far has been in preclinical systems, as it always is at this point. So as we translate into the human, we need to demonstrate that what we've achieved and what we see in our model systems translates into the human, and effectively so. And so understanding target engagement, understanding proof of mechanism in humans, demonstrating that the delta that we just talked about, that tolerability index between cancer and normal, can be achieved and sustained. Those are all the key initial components. And then, of course, showing that it actually does something to that patient's tumor, showing activity. Obviously, as we've already sort of described, we can stack the deck. We can skew these initial steps in development towards patients that are more likely to be responsive to a monotherapy MYC inhibitor. And we've already sort of outlined the indications here where we think we would initially translate. Non-small cell lung cancer, both squamous and adeno, in defined cohorts. Colorectal cancer, triple-negative breast cancer, DLBCL, diffuse large B-cell lymphoma. Those are the backgrounds where MYC is high, where we have an opportunity to see monotherapy responses and still to establish our safety, tolerability, target engagement, proof of mechanism. The question is, how can we do that efficiently and effectively? Well, it turns out from a provenance perspective, some of the initial work on this program was actually conducted in Australia. We're very proud of our Australian roots. We maintain an active group in Australia. We have an active operating subsidiary in Australia. And we have fantastic relationships within the Australian ecosystem. And Australia is a jurisdiction I think many people are starting to become attuned to, in which the efficiency of the regulatory process can be exploited to efficiently move into clinical studies and to establish some of these initial signals. We're very strongly considering initial translation of this project into Australia for the efficiency of time and the efficiency of signal generation, and then to bring in the US as well, obviously, file an IND, bring in the US, use US sites and Australian sites together, both to complete dose escalation, backfill cohorts to better understand the signals that we're seeing, to understand dose, to get to a dose, and then to think about, okay, what do we do now in our expansion cohorts? And then to open up expansion cohorts that clearly leverage MYC biology, both as monotherapy and potentially to begin to answer some of these questions around combination opportunities as well. So as we think about expansion cohorts, again, I think the question is, how do you efficiently and effectively move towards signal? We're thinking in global terms. Perhaps there's opportunities to enroll those expansion cohorts in Western jurisdictions, but perhaps there's also opportunities to enroll those expansion cohorts in other jurisdictions, for example, in China. There's a lot of discussion these days about Chinese-generated data, early clinical data coming out of China, bringing those assets over to North America and Europe. But the same opportunities exist for companies like mine to go into those jurisdictions and to generate the same kind of signal-seeking data very efficiently, very effectively, as a Chinese local company can. The story of MycRx is one of virtual development. We have no wet lab capability. All of the work that we've achieved, all of these breakthroughs on MYC, have been achieved through a distributed model. We have the internal expertise. We're all virtual. And we've directed work through external CROs throughout the world, including in places like China and India, Europe, and North America. And so we're very used to working on a global scale. We're very used to working internationally. In fact, we have fantastic relationships in China already. And so as we think about development, we're really agnostic to where we do work. What's critically important, that it's high quality work, it's GCP, that the investigators are enthused and motivated, that they're thinking thoughtfully about the patients to enroll in the study, and that we can move effectively and efficiently to generating the signals that we think could add value in the program, and to do that in an agnostic way. And I think that's the modern oncology rule, is it's all about data, high quality data, but efficient, effective. Capital resources are always challenging for biotech companies. So how you thoughtfully deploy your resources to get to the outputs that you want, I think you need to be creative. We've been creative throughout this project to this point in time. And as we move into development, I think we'll continue to be creative in terms of driving effectively and efficiently to generating those signals that we think will be value-adding for our investors, our shareholders, and obviously set up what we think could be a very important potential new therapeutic class for its next steps.
Nick, you mentioned it's important to really understand this is not only the science, but also the business of drug development. You have really been successful in both, and you have explained the science. You're very engaged in the science very deeply, but also you have a great track record in taking drug from early stage and to next milestone and exits. What wisdom you can share with someone trying to develop new drugs, new mechanism of action, and in this complex world, maybe lesson learned, some advices that you would have for a CEO of a biotech doing early phase oncology assets?
I think for small co-biotech, small co-private biotech like MycRx, the goal is exit. The question is, at what point in time is there a sufficiently compelling data set? You can't obviously build a company to sell, you have to build a company. So you're always thinking about what's the next steps in terms of building my company. But the orientation to that next step is to generate data that has the potential to be catalytic. Clearly, you don't know whether or not that data is going to be catalytic. That's out of your hands. But what you can do, and what I would strongly encourage, is to be socializing your story with those folks who at the end of the day you're hoping would be in a position at some point to potentially acquire your company, partner with your company, license your drug. We've been socializing our story with Big Pharma for an extended period of time. I think it's really important that they get to understand what it is you're doing, philosophically what you're trying to achieve and why, what the data needs to look like in order to reinforce that you are indeed achieving what you set out to achieve, to let people know what you intend to achieve by when. If you say, by this time, we will have done the following things, go off, achieve those things, come back and say, look, we did exactly what we said we would do, here's the data, here's what we're going to do next, and continue to execute and operate at a high level and drive towards the delineation of value-creating outcomes that you just haven't conceived of yourself in a vacuum, that you've already socialized with the other side of the table would be meaningful. That you know if you achieved X, Y, and Z, that the other side of the table is going to consider that important and valuable and meaningful. And of course, at some point, hopefully, if the data go your way, potentially transactable. So I guess the advice is always think from the potential exit perspective. You can't guarantee it, but try and understand what are the components in your data that could be catalytic with respect to achieving exit. Be sure that you've got your checks and balances in place, and then achieve it. Set out to achieve the data effectively and efficiently in both time and money. Deliver on the data. Hopefully the data go your way if you've designed your studies appropriately, if you've got the appropriate controls, if you've executed in a high quality way, and obviously continue to do that in an iterative manner, allocating capital thoughtfully, and getting to a point at which it becomes ever more increasingly persuasive. It's a business of persuasion, and data persuades. And as we see in ASCO, as things move further into development and more data are unearthed, things become ever more persuasive. And it's no different starting on that journey. The beginning of that journey was, can you persuade me that you actually can directly drug MYC? Well, I think we can persuade anyone that we directly drug MYC because we have very persuasive and compelling data. Now the next questions are, okay, now that you do have that, fantastic, we're impressed. Now what are we going to do in terms of showing that MYC in the clinic is safe, well tolerated, and does something meaningful? And that's the next thing we need to do. And then the next thing to do would be, okay, you've shown that, how are we going to get the drug registered? Where are we going to get it registered? How are we going to build the commercial component of drugging MYC effectively? And that will be the next step. And that's where your expansion cohorts start to set up your potential paths to registration.
What final thoughts you want to leave the audience with regarding MYC and MycRx?
The overarching philosophy that I have is, and I look around ASCO, and I've mentioned this to you personally, I believe we need to continue to break new mechanisms. It's critically important for cancer patients that we continue to break through new mechanisms that have the potential to do something different. And it's only by breaking new mechanisms can we really bring cancer care forward for the people that desperately need us to. I think it's important to stand behind the convictions that you have, to generate data that obviously continues to build your conviction, but to maintain that North Star of trying to help people who desperately need that help still. We have a long way to go in cancer. I think for a target like MYC, the opportunity to help many people is incredibly motivating. It's motivated each and every one of us. It's been an incredibly difficult target, biologically incredibly complex, very technically challenging at every step of the way. But we've persevered and we've got to this point. And now we're at the juncture where we can start thinking about actually seeing what this does in patients. Is incredibly motivating to be moving towards patient care. I think it's important that we all in this field continue to focus on what really matters, which is the patient. I've spoken about business. Perhaps it sounds like the orientation is very divorced from the patient. Nothing could be further from the truth. Everything we've done at MycRx has been focused on trying to help people with cancer as much as possible, recognizing, of course, that we are in a business and needing to understand how you bring those two worlds together in a Venn diagram overlap in which you can focus on trying to help people with cancer, achieve the business ends that you need to achieve as a for-profit entity with investors and shareholders and stakeholders, and be able to walk away saying, I was able to achieve the business ends that I wanted, but more importantly, I can see a legacy of patients who have benefited from the efforts I've put in. And that's really what I've been about my entire career. I can look around booths here and see where directly those efforts have paid off, and cancer patients are benefiting from the effort that I've put in. And certainly, as we move MYC forward, the prospect to do that at a level I couldn't have dreamed about 10 years ago, because the technology, the tools, the capabilities simply weren't there to be able to tackle a target like MYC in the way that we can today, is incredibly motivating, and we look forward to working with companies like Syneos to bring this to patients and hopefully to market.
Nick, thank you so much for the insightful conversation, and I really hope that you, Nick, will break through MYC through MycRx and help a lot of patients.
Thanks so much.
That's all for today's episode of the Syneos Health podcast, Early Signals. I'm your host, Dr. Wael Harb. If you have other topics you would like to hear on this podcast, please send us a message at podcast@syneoshealth.com. For access to more future-focused, actionable life science insights, please visit the Insights Hub at syneoshealth.com.